learn object concepts. And then from there, he went on to do a postdoc at Carnegie Mellon University, working with Dr. Marlene Behrman. There, he really built on this line of work that he established in his PhD, now investigating the roles of different visual pathways for their support of object perception, particularly in patient populations, and looking at the capacity of these pathways for reorganization following massive surgical resections. Central to this really already impressive body of work is the question of how humans acquire new visual concepts with minimal or really no direct experience. And I say it's been a delight working with Vlad these first couple of months. And in the lab, he's probing the brain's organization at birth and assessing how it provides a scaffold for these abilities to really form and emerge later in childhood. I'll say one other thing. One of the aspects of Vlad's research that I think truly stands out is the extensive array of behavioral, computational, and neuroimaging techniques that he employs to really dive deeply in addressing these developmental questions. And I believe this will really be evident in today's presentation, where he'll discuss the early developing mechanisms underlying visual object categorization. So please welcome Vlad.
Well, thanks, Mike. Can everybody hear me okay? And then I know there's people on Zoom, so sometimes I might wander. So Heather, let me know if people can't hear me. Yeah, so title of my talk is Early Developing Mechanisms Underlying Visual Object Categorization. So the human visual system has a remarkable capacity for abstraction. For instance, this 10,000-year-old cave painting bears few properties of real-world objects, but even a child could tell you that it shows a bunch of people and animals. And so we know that our visual systems are extremely powerful and are able to interpret visual information quickly and effortlessly. So as an example, here I'm showing you objects presented for just 70 milliseconds each, so less than 1/10 of a second. And yet it's trivially easy for us to identify these objects as they appear. And as the cave paintings illustrated, the specific input needed to recognize the object need not bear any resemblance to the real world and maybe hasn't even hit your retina before. For example, you've probably never seen a wavy lamp or a wavy airplane, but we can still recognize these objects even at this speed. In fact, I can show you an object that you've probably never seen before, like this instrument called a sheng, and you would readily be able to identify other examples of a sheng and do so across many contexts. So how do we do this? How do we make these kinds of generalizations with so little experience? So this is the broad question that my work seeks to address. And specifically, I'm interested in understanding the computational and neural mechanisms that support such categorization abilities, and critically, how they might arise over developmental time, starting in infancy. And as I've kind of alluded, I primarily focused on this question in the domain of object categorization. That is how we might recognize an object we haven't seen before. But this question is also important for understanding how humans rapidly acquire other kinds of cognitive abilities early in development. But as a starting point, today I'll tell you how I've begun to address this question in the context of object categorization. So let me give you a little bit of a roadmap. So I'm going to start by examining what kinds of visual representations might support object categorization at the earliest stages of life in infancy. And then I'll ask what neural mechanisms might support such object categorization abilities, at least in the mature system in adults. And then finally, I'll integrate this developmental and neural question to ask, how do the neural pathways underlying object categorization actually develop? So to start with the first question, what visual representations support categorization in infancy? Well, to go back to the beginning of the talk, we know that we can identify objects even when they have few properties of the real world, such as these cave paintings. And we can recognize exemplars of a category even when they are new and have very few local visual features in common. So this suggests that we don't rely on any specific visual features to recognize an object, but might categorize objects on the basis of some more abstract object property. So what might that property be? Well, one possibility is that we form such object representations not by using local features, but by using their spatial relations. That is, how those features are arranged. So consider a common set of objects such as these guitars. So these guitars all have different local visual features, but the spatial arrangement of those features, so where the neck and body of the guitar are relative to one another, is very similar. In fact, I can almost entirely remove those visual features, and you can still recognize this object as having a guitar shape. But if I simply rearrange those features, it becomes a totally different object. And this might feel reminiscent of how David Marr initially described human shape deception, not as a description of any specific contours or surfaces of an object, but as a structural description that describes where those features are relative to one another. And so we can see how spatial information could be used to form an abstract shape representation. But now we might ask, how do we actually test whether infants use this kind of information to categorize objects? Well, this is where computational models can really help us. Specifically, we can implement different visual representations into computational models and then test which models best explain behavioral and neural data from humans. And so here we tested whether a model that describes an object's spatial relations does a good job of fitting human categorization abilities. And specifically, we tested what's known as a skeletal model shape or shape skeleton. So shape skeletons are pretty intuitive. One can think about them as kind of like the stick figure of an object. You know, a stick figure provides a good descriptor of the human form without needing to describe all the surface properties of a person. And so shape skeletons might provide a good description of objects more generally across different kinds of variations. And as I've already alluded, what shape skeletons describe is the shape topology, that is the spatial arrangement of object features. So where the arms and legs are relative to one another on a person, or where the wings and body are relative to one another on an airplane. And crucially, shape skeletons stay stable across variations in category exemplars, such that again, even though these guitars have different local features, their underlying skeletons are very similar. And really what skeletons give you in this context is a common format by which to compute shape similarity. So even though the skeletons of these guitars are a little different from one another, they're still more similar to each other than they are to any other object category. And so we can see how we might use shape skeletons to accomplish object categorization. And so now the question is, do infants do this? Do they rely on a skeletal representation to categorize novel objects? I'm going to try not to knock this over during the talk. All right, so to test this, we recruited 84 6 to 12-month-olds. And we specifically tested whether infants categorize novel objects by their skeletons across the changes to their local visual features. So you can see that these two objects have the same skeletal structure, but their local visual features are different. And we specifically chose this age range because we know that infants have very little object experience at this age. In fact, just 10 objects occupies about a third of their visual experience. So we know that these objects are going to be totally new to infants, and they shouldn't really have any expectations about how these objects should vary within a category. And so we tested whether infants correctly categorize these objects, and we compared them to a series of other visually matched objects. And so to give you a little bit more detail about the procedure, we used what's known as a habituation-dishabituation paradigm. So infants were shown one object over and over until their looking time decreased to criterion. Effectively, they got sick of looking at this object. And then we showed them one of two new objects. One that had a familiar skeleton, and then one that had a novel skeleton. And as you can see, both objects now have different surface features, different local visual features. And so the prediction is that if infants generalize across a change to the object's local feature, they would look longer at the object with the novel skeleton, but not longer at the object with the familiar skeleton relative to the situation. And importantly, we made sure that infants can equally discriminate between these objects and the habituation object, meaning that we made sure that categorization here wasn't simply because they couldn't see this different local feature. And so we tested infants in two experiments, one where the object structures were relatively different from one another, and one where they were much more closely matched. Indeed, you can see that the image similarity between the second set of objects is much more similar. They're much more similar on the retina. And so what we found first is that infants could generalize across a change to the local feature. That is, they did not dishabituate to the object with familiar skeleton and only dishabituate to the object with the novel skeleton. And this is a finding that we replicated in the second experiment where those objects were much more similar to each other. And so this again suggests that infants can generalize across a change to the object's local feature, but we might still ask, how is this accomplished? And they really categorize these objects by relying on a skeletal structure. And so, as I mentioned, we also compared infants to a skeletal model, and as a control, we compared them to a series of deep neural network models. So some of you may already be familiar with deep neural networks, but basically deep neural networks are computational models that learn to categorize objects after being exposed to many training examples. And the reason why people are so excited about deep neural nets is they can do a lot of the same tasks humans can do using similar inputs and outputs. Meaning you can show a deep net, a naturalistic image, and then they can classify the image into some number of categories. And what makes them particularly interesting from a neuroscience perspective is that there seems to be a functional correspondence between the layers of a deep net and the different layers of the primate ventral visual pathway, the pathway that supports object recognition. And so we tested infants against all these models, and we're pretty careful about how we chose these models. Specifically, we compared infants to a model with a more biologically plausible recurrent architecture, such as CoreNet-S, a model whose training was specifically intended to induce a shape bias by decorrelating color and texture information, and a model that was trained on naturalistic headcam videos from infants. And so even though the neural net literature moves pretty quickly, these are still some of the best models of human vision and object recognition. And we might, reasonably predict that these models should at least have the recognition abilities of a 6-month-old. But since we know that these models don't compute a shaped skeleton, we also thought they would provide a really stringent test. And importantly, we also tested these models in a way that was comparable to infants, specifically by converting each model into an autoencoder. So an autoencoder is simply a model that recreates its input from some lower dimensional space, here from the penultimate layer of these models. And the reason we did this is that one can conceptualize looking time in our task as a measure of alignment between the stimulus and the internal representation and the infant's internal, such that infants will continue to look longer at a stimulus that is unfamiliar compared to one that's familiar. And so similarly, the error signal of an autoencoder will also remain high when there's a mismatch between the internal representation of the model and the original input. And so what this means is that we could habituate the models using the same videos infants saw, and then test the models using the same videos infants saw. And so we compared infants against all these models, and we converted all their scores into a categorization score, which is just a proportion of looking to novel versus habituation. And what we found is that, again, infants could generalize across a local feature, and their performance was best matched by a skeletal model, not by any of the other deep neural networks. And this is something we also found in experiment 2, where the deep nets didn't even perform above chance. And so these findings suggest that infants categorize these novel objects by representing their skeletal structure.
Really?
Yeah, Russell. Yeah, and also I'm open to questions in general.
I guess one might try to understand. I just thought you were going to do something about how representation is similar.
Yeah.
Or I guess that would be indexed in the extent to which they can recreate the image, but actually you built in some annotation into the models. Yes.
So just kind of rephrase your question a little bit. Yeah. So one common way of comparing humans to models is using representative similarity, basically asking whether the models represent stimuli as in a similar way to humans, right? That they think two objects are the same two objects are the same that humans think. So typically with adults, you need a lot of trials to do that, which unfortunately with infants, we have like 10 minutes of data collection. So we can't quite get the trials to do that. So here we ask whether the performance was the same. Given these controlled conditions, which models show at least the same kind of behavioral performance under as matched conditions as we could create, which here we were able to approximate a habituation dishabituation paradigm in the models. Does that answer your question?
So you're actually looking at some sort of change in the models,
So
learning within the model.
Yeah, so the model weights themselves are fixed.
Yeah.
And the only thing that changes is the task. So now you add an additional layer, the decoding layer, right? And you just ask, given the existing representation of the models, would it also habituate and dishabituate the same way infants do, right? And so the only thing that changes is it learns that habituation video, and we use the same criteria we did for infants. So we pass that video through, we measure that error signal goes down the longer we pass that video through, so it meets the same criteria infants met, and then we send the test videos through and ask whether their performance was similar or dissimilar to the infants. So the only training is basically the only training infants got, which is the habituation, is the task itself.
I guess the weights were fixed.
The weights of the model are fixed, and then there's a decoding layer.
Decoding.
Yeah, and only that's, yeah. So it's as if, you think about infant comes in with whatever representation it comes in with to the study, right? And then it learns something during the task, which is just the habituation, right? And then it has to make some inference. Then the baby has to make some inference from the basis of that habituation. And it was the same thing with the models, where the visual representations of the models, the encoder, were fixed, and then only this decoding layer was trained using only the habituation video. Yeah. Any other questions? Cool. Yeah, I should have paused at the autoencoder because it's definitely a different approach than we typically use
I
for.
think I still entirely get it. I don't want to play down.
Sure. Okay. Okay. So we found that infants categorize novel objects by representing a skeletal structure. But we might ask, why did the deep neural nets perform so poorly? These are supposedly the best models of human vision. Maybe there was something weird about our stimuli or this testing procedure that we used, right? This is a little bit unconventional. So to examine this possibility, we tested how well these models categorize objects by their local feature here across the skeleton change. So the models now were habituated to one object and then tested with objects that either had the same local visual feature or a different local visual feature. But now the skeletons are different from habituation to test. And now in this case, we found that the deep neural networks, but not the skeletal model, could categorize the objects using their local features. So this suggests that our results weren't just because of something weird in our testing procedure, but instead, infants and neural networks might use qualitatively different kinds of visual information to categorize the objects. Yeah.
Has the baby study been run of this where you can habituate them?
No, so we have not run this version, and unfortunately, a lot of this is due to just time and resource constraints. Yeah, collecting 80 for infants for even the first study takes forever. But yeah, I would love to run this version. And then there's other kinds of variations of this that I think would also be really valuable to run.
Then what's your prediction of what babies?
So we already know from the pre-testing that we did that infants can see the difference between these features, right? They can discriminate these just fine. The question that's open is whether they would categorize them, right? So beyond just visual discrimination, would they use this as a diagnostic cue to form some category based on that? I'm not sure. I think given other literature, we know that infants have a shape bias, which is essentially a structure bias. So if we were to pit them against each other, one would predict that they would still use the skeleton over the local feature. But in other contexts where you may say you vary the skeleton, so it's uninformative, they could learn to use the feature as well. Yeah. Any other questions here? All right, so this finding that deep neural nets could categorize by local features, but not the skeleton, is actually really consistent with other accumulating evidence that deep neural nets might not represent shape at all. For example, these models are frequently misled by a simple perturbation to the object's contour. So here predicting that this image is more likely to be a parachute than a hammer. And instead, they seem to rely on very local visual features. For example, this image contains the rearranged pieces of a hammer, but no human would label this as a hammer. But the neural nets, label this as a hammer with about as much confidence as they labeled the other one a parachute. And perhaps most famously, we know that they're affected by so-called adversarial attacks, where a tiny image artifact completely changes the predicted label of the object. So here calling this baseball a green lizard. And so to briefly summarize, we found that infants can categorize novel objects across changes to their local visual features. These are objects the infants haven't seen before. And their categorization performance was best matched by a skeletal model rather than various deep neural networks. And by contrast, the deep neural nets were better at categorizing objects by their local visual features. And so together, this suggests that shape skeletons may be one mechanism by which infants rapidly learn object categories in the absence of extensive experience. And so we found that one visual representation that might support object categorization in infancy is a skeletal representation of shape. And so now we might ask what neural mechanisms support such object categorization abilities, at least in adults. And so decades of research has shown that object recognition is primarily supported by a hierarchy of regions along the ventral visual pathway. But as I've already shown you, the best computational models of the ventral pathway, deep neural networks, may be missing a crucial aspect of object recognition, namely a representation of the shape structure. So obviously, we shouldn't assume the biology is wrong just because we have a bad model. But in this case, there's reason to think that the deep neural nets are actually a lot more like the ventral pathway is actually a lot more like the deep neural nets than we even previously believed. In fact, there's accumulating evidence that the ventral pathway may also not compute shape, but may be better described as a basis set of local features. For example, there's evidence that a slight perturbation to an image can radically change the firing rate of ventral neurons. So if you take this image that preferentially responds to pipe gauges and not dogs, and then add just a small image perturbation, much like an adversarial attack, you can radically change the firing rate of that neuron such that it now responds even more to these images of dogs than it initially did to pipe gauges. And you find converging evidence at the population coding level. For example, if you present participants with images, whose spatial arrangements are intact or scrambled, behaviorally, humans can readily discriminate between these images, meaning that they can readily tell the difference between these intact ones and these scrambled ones. But the representations of the ventral pathway, as well as deep nets, treat all these equally, suggesting they cannot discriminate between intact and spatially scrambled images. And so, if the ventral pathway isn't sufficient to compute shape information, then what are we missing? One possibility is that a global shape structure, like a skeleton, may be computed in regions of the dorsal visual pathway, such as the interparietal sulcus or IPS. And then this information may be transmitted to the ventral pathway to support object recognition. And so why exactly do we think the dorsal pathway plays this role? Well, from classic research, we know that the dorsal pathway plays a key role in computing spatial relations more generally. And so one hypothesis is that the dorsal pathway may also compute the spatial relation between object features to form a kind of a global shape representation. So I tested this hypothesis in two experiments using fMRI. And we first tested whether the dorsal pathway represents the spatial relations between object features. And then we asked whether these regions actually contribute to object categorization. And so for the first study, I created a functional localizer task to identify regions that are sensitive to an object spatial relations. So here, participants were shown blocks of trials where either the spatial arrangement of features varied from trial to trial, while the features themselves stayed the same. So you can see on the left, all those images have this hourglass shapes, but those arrangements of features where that hourglass are relative to one another changes. And we contrasted that with blocks of trials where the features vary from trial to trial. So circle, square, whatever, but the arrangements stay the same. So they might all have this T arrangement. And here participants simply did a one-back task. And so using this contrast, we found two regions in the dorsal pathway that were more sensitive to the arrangement of features than the appearance of features. One in more posterior IPS and one in more anterior IPS. And at the group level, we only found these regions in the right hemisphere. And importantly, these results were pretty consistent across participants. In fact, every single participant, I don't know if you can see my mouse here. Yeah. Every single participant showed these regions in their right hemisphere, but you can see that it's a little less consistent in the left. Yeah.
I mean, but I just clarify, you do get it.
You do get it. Yeah. And I analyzed both hemispheres in every case.
No, not quite. Ventral, you do have ventral stream.
Yeah. And I'm going to test that as well. Yeah. Don't worry. I'm not ignoring anything. Yeah. But yeah, so we do see it in both. And then we're going to test where it's really originating from. I promise I'll get there. Any questions at this point?
Where are they control?
Yeah, so they're not really IT. So they're actually more middle temporal.
It's like the lateral part of LO.
Yeah, something like that. Yep. Yeah. I'll come back to it, I promise. So having found these regions in the dorsal pathway that are sensitive to an object spatial relations, we next examined the selectivity of these regions. And we did this by comparing this object relations localizer to other properties we know the dorsal pathway is sensitive to, such as allocentric relations. So here participants had to judge whether the distance between this circle and line were the same or different across these mini displays, and that was contrasted with this brightness judgment task. We also compared them to a depth localizer, which contrasted 3D shape from 2D shape, as well as an action affordance localizer, which contrasted tools from manipulable non-tool objects. And so what we found in held out data is that overall, these regions responded more to the object relations condition more than the other properties we know the dorsal pathway is sensitive to. And these effects were strongest in the right hemisphere. And I just want to note, so I don't have these graphs here, even though those regions showed a greater spatial greater than feature response, there was no selectivity. relative to these other properties. And then stick with me because another experiment I'm then going to ask about directionality. But anyways, this suggests that there's selectivity for relations in these regions over and above these other properties in the dorsal pathway. So having found regions in dorsal pathway that are sensitive to the spatial arrangement of features, we next tested whether these regions actually contribute to object categorization. And so here we simply presented participants with 5 categories, each with 5 exemplars. And there was no special task here. Participants are simply shown a stream of object images, one after the other. And the first thing we want to know is whether we could decode an object's category from these regions. That is, if we trained a classifier on the neural representation for 3 of these lamps, would it also classify this held out example as being a lamp and not say it is one? And what we found is that we could decode an object's category only in right posterior IPS, not any of the other dorsal regions. But now we might ask, how does this decoding compare to the ventral pathway? So we repeated these analyses using a ventral object region here at the lateral occipital cortex. And perhaps unsurprisingly, we also found that decoding was above chance here and not significantly different from posterior IPS. But now we might ask, how is decoding being accomplished here? Are dorsal and ventral regions using the same kinds of information to categorize objects? So here again, we can use models to explore what kinds of visual information these regions may be using. So if these dorsal regions are categorizing objects by their spatial relations, we might predict that the response profile should be well captured by a model that describes the spatial relations among features, namely a skeletal model. So to do this, we computed the dissimilarity between each of our objects using the skeletal model, here shown in this matrix. And so blue cells in this matrix, I don't know if you guys can see that, blue cells are object pairs with more similar skeletons, and then red cells are object pairs with more dissimilar skeletons. And so essentially, this is our prediction space for how similar or dissimilar brain regions should see these objects. And so as a control, we also included other models of vision, namely CoreNet-S, which is a deep neural network that approximates the ventral pathway, as well as the Gabor-Jet model, which approximates early visual areas. And so the approach here is to conduct linear regression analyses with each region as the dependent variable and each of these models as the independent variable, and then examine the unique variance explained by each of these models. And so what we found is that the skeletal model was only predictive in right posterior IPS, not in any of the other dorsal regions, which again is consistent with our decoding results that this was the only region with above chance decoding. But we also found that the skeletal model explained significant variance in left LOC, a ventral area alongside a deep neural network. I'm sorry, a ventral area, and here it explained variance alongside a deep neural network. And so now we have evidence of spatial relations being important for both areas. So does that mean that part relations arise in LOC via input from IPS? Could it be the other way around? Or maybe this information is just being computed in parallel. And so to test this possibility, we conducted mediation analyses. Specifically, we tested whether the relation between the skeletal model and left LOC could be explained by the response profile of IPS. And what we found is that IPS fully mediated this relation, meaning the variance explained by a skeletal model in LOC was fully accounted for by the response profile of IPS. And the reverse was not true. And so this provides some initial evidence that skeletal representations in ventral areas, in regions like LOC, might arise by way of IPS. However, as many of you may know, fMRI has very low temporal resolution, and so it's hard to make firm conclusions about the directionality of some of these interactions. And so in a subsequent study, we used high density EEG, which has high temporal resolution and sufficient spatial resolution to dissociate parietal and temporal lobes. And what we found is that category information could be decoded earlier in the dorsal pathway than the ventral pathway. So here I'm showing you decoding accuracy as a function of time for the two pathways. And indeed, across many resamples of the data, we found that decoding was consistently earlier in the dorsal pathway than the ventral pathway. So here I'm showing you the frequency of first decoding in each of the two pathways. And importantly, we found that the dorsal pathway also predicted the responses of the ventral pathway. So this is a time generalization matrix showing you the correlation between every time point of the dorsal pathway with every time point of the ventral pathway. And as you can see, the correlations are generally stronger under the diagonal where dorsal predicts future time points of ventral. In fact, if we rank order these correlations, we can see that the majority of top correlations are those where dorsal predicts ventral rather than the other way around. And so together, these findings suggest that structural information like a skeleton may arise in the ventral pathway by way of the dorsal pathway. And so to provide a brief summary, we found the dorsal pathway contains regions with selectivity for object spatial relations, which may allow it to compute an object structure. Importantly, the representations within these regions was capable of supporting object categorization. And finally, our fMRI and EEG data suggest that the dorsal pathway may actually transmit this information to the ventral pathway rather than the other way around. And we've actually formulated a more general framework to synthesize all these findings, which we describe in a recent TICS paper. So there's a lot more to this opinion paper that I can get into now, but essentially we drew on evidence from different kinds of neuroimaging modalities, modeling, and patient work to argue that the ventral pathway might be better described as a basis set of local features, much like the feature space of a deep neural network, and that global shape properties like the skeleton may arise by way of the dorsal pathway. And so we suggest that a complete object representation, one that integrates structural information with features, arises via the interactions of these two pathways. And I just want to note that this framework is not limited to just objects and may hold for other categories such as faces, and actually maybe scenes too. Indeed, there's evidence that high-level face regions actually are extremely sensitive to local visual features more than kind of the entire face. And instead, spatial mechanisms in the dorsal pathway might be needed to describe how those facial features are actually arranged in a face. So basically face configuration. And so we found that the dorsal pathway may actually compute an object structure and support categorization. So now I've shown you that categorization in infants might be supported by a skeletal representation of shape, and that such information may be computed by the dorsal pathway. And so a reasonable hypothesis is that the dorsal pathway may also develop early to support these kinds of representations in children. But we haven't actually tested this yet. And so in the final section, if I can move on. Oop, jumped ahead. So in the final section, we asked, how do the neural pathways underlying object categorization develop? And specifically, what is the developmental trajectory of the dorsal pathway? And might it even develop earlier than the ventral pathway? So let me show you how I've begun to test this question. So if the dorsal pathway develops earlier than the ventral pathway, then one prediction is that the network organization of the dorsal pathway will be more adult-like than the ventral pathway, even in newborn infants. So I've begun to test this question with Mike using newborn fMRI data where infants were scanned while they're asleep. And again, these are newborn infants that were scanned on their first day of life. And so to do this, we registered visual maps from the adult brain to the infants, and I can get into more details of how this was done. And then we computed the resting state functional connectivity correlations between 25 visual areas in newborn infants as well as adults. And we can see these in these chord plots where each line in these plots is a time series correlation between two visual areas. And what we found was that the pattern of connectivity in infants already showed evidence of an adult-like organization. So to visualize this another way, we can plot these similarities in 2D space, and we can see distinct clusters for early visual cortex here in black, dorsal pathway in red, as well as ventral and lateral pathways. And this organization is overall really similar to adults. And so I just want to take a second to step back and just appreciate just how similar the organization of the infant brain already is to adults, meaning that even at birth, the organization of the visual system might be largely in place. But importantly, we can now ask how adult-like are each of these networks? And is the dorsal pathway already more adult-like at birth than the ventral pathway? And what we found is that yes, the pattern of connectivity of the dorsal pathway is more adult-like than the ventral and lateral pathways. So this provides some additional evidence that even at the earliest stage of life, on day one, the dorsal pathway may be more mature than the ventral pathway. And so I'll just pause here in case there are questions. Yeah. And so we found that the network organization of the dorsal pathway may develop early. And this finding leads to another really interesting prediction. Specifically, if the dorsal pathway does develop earlier than the ventral pathway, then dorsal pathway may also show less functional reorganization than the ventral pathway after brain surgery in childhood. So let me unpack this a little bit. So we know that the brains of children are generally more plastic and adaptable than adults because they're still immature. But as I already showed you, the entire brain doesn't mature all at once. So if there are regional differences in the development of different pathways, we might predict that these same regions might be more or less able to adapt following damage or surgery. So I've had the opportunity to test this question in a really rare group of patients that had hemispherectomies as children. So basically, these children had entire hemispheres of their brain removed to treat drug-resistant epilepsy. And so before I continue, I just want to note that these kids are amazing. If you met them, you would not know that they are missing half their brains. They score very high on most cognitive tasks. They obviously have muscle weakness on the contralateral hemisphere, and they're blind in one hemifield, but a lot of them are in college, they ride horses. It's actually truly remarkable. But this group also gives us a really rare opportunity to design an experiment. So let me tell you a little bit more about what we did. So one property of the brain is that some functions are lateralized. So most people know that language is primarily lateralized to the left hemisphere. And as a result, the representations for things like words is also primarily lateralized to the left hemisphere of the ventral pathway. And by contrast, the representations for things like faces is primarily lateralized to the right hemisphere of the ventral pathway. And as I've already shown you, there's also lateralization in the dorsal pathway, such that things like global form seems to represent more strongly in the right hemisphere of the dorsal pathway. And extensive literature has shown that things like tools are represented more strongly in the left hemisphere of the dorsal pathway. And so now we might ask, what would happen if we lose one of these areas in childhood? What if you were to lose an entire hemisphere? Would the functions of the ventral and dorsal pathway reorganize to the contralateral hemisphere? So we tested this question with an equal number of left and right hemispherectomy patients. So first, I want to make some predictions for how words, faces, tools, and global form might be represented in these patients. And as a note, any data I show will be from patients' intact hemisphere, meaning that pink will always be data from a patient's intact left hemisphere, and blue will always be data from a patient's intact right hemisphere. Otherwise, it gets confusing which hemisphere is lost, which hemisphere, et cetera. So for words and faces and controls, we should see that the response that there should be more selectivity to words in control left hemisphere and more selectivity for faces in control right hemisphere. This is the classic lateralization effect. And if the ventral pathway matures fairly late, then in patients with a missing left hemisphere but intact right hemisphere, we should see reorganization for words such that their response to words is now on par in their right hemisphere to control's left hemisphere. And by contrast, we might see something similar for faces, such that the response to faces in patients' intact left is now on par with control's right hemisphere. And we might otherwise see normal responses to these categories for those intact hemispheres, given that they might not otherwise be disrupted. Now, for the dorsal representations in controls, we should see that classic lateralization effect, where tools are represented more strongly on the left and global forms are represented more strongly on the right. And mostly normal responses for those categories for patients that still have those specific hemispheres. But if the dorsal pathway matures earlier than the ventral pathway, we might not see evidence of reorganization of function to the contralateral hemisphere. Does that make sense? Yeah, Sarah.
So you predicted increase in selectivity in the hemisphere that doesn't usually handle that in the hemispheric radiation, but why do you predict a decrease in selectivity for like language in the left side?
I don't. Yeah. Oh, yeah. It's, I guess, slightly lower. It's these patients have lost half a brain, so this is just a schematic, and the data are never as clean as this. But obviously, I'll show it to you in just 1 second. But yeah, I felt too ambitious to say it'd be exactly the same, so I gave it a slight nudge down. But yeah, I should probably just change that so that it's not. I'm not making a specific prediction there. But yeah. Any other questions here? Or about hemispherectomy in general? It's a very cool population. Okay, so now for the results. So first, if we look at the ventral categories for controls, we see the classic word-face distinction, such that in controls, words are represented more strongly in the left hemisphere, faces are represented more strongly in the right hemisphere. And so to orient you to these figures a little bit, this metric, sum selectivity, is just a measure that takes into account both the strength of activation and the total amount of volume active. So it's a composite score. And because we only have a few patients, we use non-parametric statistics. So these violin plots are the bootstrap distribution of control data after 10,000 resamples. And so now, if we look at the patient data, we see evidence of reorganization of the ventral pathway, such that the majority of patients with a right hemispherectomy now show normal responses to words in their intact left hemisphere. And similarly, we found that patients with a right hemispherectomy now show a normal response to faces in their intact left hemisphere. And so this suggests that there's some reorganization of function in the ventral pathway such that those functions reorganize to the contralateral hemisphere. But the story for the dorsal pathway is a little different. So first, in controls, we again find the classic lateralization effect such that tools are represented more strongly on the left and global form is represented more strongly on the right. But we see very little evidence of reorganization in patients, with only about one showing any kind of normal response to either condition in comparison to any hemisphere. So together, this suggests that the dorsal pathway exhibits less functional reorganization than the ventral pathway, perhaps because the dorsal pathway matures earlier than the ventral pathway. Do you have a question?
Global form.
Sorry?
Global form.
What about it?
You haven't checked.
Oh my gosh, wow.
Yeah,
There you go.
great.
Sorry about that. I thought they both come up at once. That was gonna be a big reveal. So yeah, same general pattern. Okay, so together these findings provide converging results that the dorsal pathway may mature earlier than the ventral pathway. And so, how do the neural pathways underlying object categorization develop? Well, I've shown you that the dorsal pathway may be early developing, at least relative to the ventral pathway. And so to summarize, using classic methods from developmental psychology with computational modeling, I showed you that rapid object categorization in infancy may be supported by a skeletal representation of shape. And then using data from adults, I showed you that this kind of representation may be actually computed by the dorsal visual pathway, not the ventral pathway. And finally, I presented some evidence from both infants and patients that suggests that the dorsal pathway may mature earlier than the ventral pathway to support these kinds of abilities in children. And so to go back to these examples from the very beginning, we can imagine how we could recognize objects in these contexts by relying on a deeper representation of structure based on the shaped skeleton. Thank you. Oh, thank you.
But I guess to make the claim that like infants are really representing skeletons, doesn't it have to be true that the skeletons of things that we want to call the same object are more similar to each other than a skeleton of like one object is another object? And is that true? And like as a corollary, how do you decide what a skeleton looks like?
Yeah.
Are there principal ways that you establish like this is like joints, things like that?
Yeah. So two questions there. How basically, and if I'm interpreting your question right, there's this tension between overall similarity and the skeleton, right? Which is always really challenging because if objects in our world are organized by their skeletons, aren't they already more similar? So we can test that, and we test that both in adults and infants using a couple different ways. Basically by changing properties that are orthogonal to the skeleton. So in our case, we changed those local visual features. So we really changed the image space of the objects, kind of their similarity along pixel space, essentially. And we did a whole bunch of pre-testing to show that objects with those similar surface forms were more similar to each other in pixel space and according to other models than they were in than objects with the same skeleton but different surface features. But you're right, this is a perceptual phenomenon where if we see objects with the same skeleton more similar to each other, then we perceive them as more similar to each other. So, again, we change the objects along another dimension, and we also test whether it's really described by a skeleton model, or maybe it's described by other models that are just general models of visual similarity. And as I've kind of shown you, we can dissociate the skeleton model from these other visual models, and our perception of object similarity is better captured by a skeleton.
I guess representing skeleton, how it's developed. Is it like I continue to the extreme, then the skeleton is just going to recreate the actual object.
It recreates the structure of the object, but not its surface features as. Yeah, it depends on the higher. Yeah. And so in some work with adults, we actually tested basically how detailed that skeleton was. And that was actually also part of the motivation of the second experiment. So you can see that the structural differences here. So we basically match the qualitative similarity of those objects. They had the same qualitative structure. And if you want to get really detailed, it's like a more Biederman-like spatial relations. And then we parametrically change the position of one part. And then what we've done in adults, which is tough to do in infants, is we really, change that position in 10% increments, right? And we find a parametric function such that adults really do perceive, that the similarity between those objects parametrically as a function of skeletal change. And again, you can partial out other models to see, is it really skeletal change that's driving that versus some other visual dimension? And we find the skeletal model explains that ability. But yeah, dissociating various types of similarity is tough, but I think we've done as pretty well to at least compare other viable alternatives to how we're going to do this. And then to kind of answer your second question about how it's computed, it's a model you can download. There's a formal mathematical definition of how you compute a skeleton. There's a lot of psychophysical work, like neural nets. There's variations, like families of these models, and there's models that both we've shown and other people have shown that are the more biologically plausible version of this, and that's the model we use for all these comparisons. Yeah. I'm gonna take a question there, and I'll come back to you, Russell. Yeah.
Be implicated in like speaker representations and also relating perceptual and conceptual knowledge, which is also important.
I have not, and I don't feel like I know enough about perirhinal cortex to comment on it, but maybe we should talk more, because one of the things I am interested in is this link between perceptual and conceptual knowledge and how this might link back to memory. So maybe we should chat a little bit more about that. Yeah. Russell.
I was originally going to ask about skeleton, sort of follow up with that question, because I guess I was a little confused. You have clarified a bit, because some of your early examples seem like they were like grass fire models.
Yeah, like the original
But
below.
that's not really what you mean. What you really mean is something more like a structural model.
Yeah.
And I guess some of us are kind of curious exactly what goes into that structural model.
Yeah.
Like, is it really about the joints, and is there like, flexibility and sort of the length of things? Obviously it's specified because you're able to, there's a way to calculate the similarity between all these, but it's clear that you mean like an actual 3D structural model. Right?
Yeah, and let me comment on that a little bit more. How much detail to include in this is always a little challenging. So, and like you're saying, this is a family model, so different groups compute it differently. And we've actually, in many of these experiments, tested multiple models. We just take, okay, there's 3 ways to compute a skeleton. Let's do it all 3 ways and see whether it makes a difference. Generally, they all explain the same variance. So one way in some of my adult work and other fMRI work, we've used a 3D skeletal model, and because we use novel objects, we had ground truth of what those skeletons were, and so we can compute that similarity in 3-dimensional space. In the infant work and in the fMRI work I showed here, we actually used a 2D projection of the skeleton across whatever the object views, and that does just as well. And we actually found that in adults as well. That even, and again, this is across orientations, right? And part of this, what I think the strength of a skeletal model is that up until foreshortening, it maintains that structure, right? It maintains what that object looks like. And then of course you lose a lot of things once an object is foreshortened, right? And so I used to really think that 3D skeleton was really important, and just empirically that hasn't necessarily borne out. They generally do equally well.
Interesting. But that's not the question I want to ask.
Okay, great.
From Zoom.
Wait.
Oops, sorry.
That's fine. Go ahead. Ask the question. All right. I want to ask about the face space, because you showed you showed some area in the ventral stream where there's not sensitivity to like organization of like even the parts of the face, but my understanding is that there are some parts of the ventral stream that very much are sensitive to that. I'm thinking of dorsal, or maybe I'm misinterpreting that. So
Yeah, I would. So there's 2 tensions there. So first, I would question whether there really are sensitive to the arrangement of the face, period, right?
Okay.
And Mike can probably comment on this even far more than I can, but I think there's now good accumulating evidence that it is pretty local, even at these high-level face regions. And second, ultimately, the dorsal pathway is sending information to the ventral pathway.
Yeah.
So if we had a time course of activity, kind of like in my data here, at a later point in time, we might see that structural information, right? Because ultimately, that the ventral pathway is still doing the task of object for face recognition. So that information has to get down there at some point. But yeah, I would actually say that ventral pathway is more local feature, like it hasn't been tested carefully enough to really tease those apart systematically. And I think it's more local featured sensitive than we previously thought,
Okay.
even in those data. Yeah.
Let's turn it on.
Okay, so
Yeah.
the question is from Sabine Spounet. Got that right? Did I say that right? Thanks for an interesting talk. Here you have quite basic level categories. Do you think that generalization for more superordinate category, big categories such as animate, inanimate, would also be possible based on a skeleton shape? Because here shapes are now very much different. Right?
Yeah. So ultimately, the skeletal model is a perceptual model. It's describing shape similarity. So any category structure that maps back onto shape similarity, it will do a good job of. But yeah, at some point, our conceptual categories break down, right? So one might say that dogs and horses have a fairly similar structure, and so the skeletal model might not do a very good job associating those two. And similarly, for superordinate categories, it might actually do a very good job grouping animals together and away from furniture, given that those skeletons are different. But then we, as humans, we form really truly abstract categories that don't match directly onto perceptual similarity. Yeah.
I had a question about the hemispherectomy
Mhm.
data. It looked in the data
Mhm.
like the dorsal stream, the dorsal stream areas didn't even have the representations that you would expect. So if you damage the right, the
Yeah.
left also doesn't show tools.
That's right.
That's not, so I just wanted to make sure that
Yeah, that's
that
true.
was also your interpretation of these data. I
Yep.
don't think it changes the conclusion that
But yeah,
these areas
it's
don't seem to be overly
yeah,
organized. But
right.
it does
So-
suggest that even the attack stuff.
That's right. And I'll note that the attack stuff is not perfect in the ventral pathway either. Right. So typically in the ventral pathway, patients were similar to at least one hemispheric controls overall. Right. And then there was also evidence that they're even similar, more similar to the contralateral hemisphere. But yeah, the dorsal pathway is just disrupted all around. So there's a couple things that might be going on. There's a general dorsal vulnerability hypothesis, which suggests that so there's a lot of disorders that are linked to dorsal pathway dysfunction in like developmental disorders. So Fragile X syndrome, Williams syndrome, a number of visual disorders. And so there's a general hypothesis that the dorsal pathway might generally be more vulnerable to any kind of disruption. And so one thing to keep in mind is that before these kids had hemispherectomies, they also had really bad seizures, right? And that epilepsy might have already started to cause tons of global disruptions beyond just, in the hemisphere. And so one of the questions that we grapple with the paper is this really reorganization in response to the hemispherectomy, or did the ventral pathway already start to reorganize to compensate for the epileptic hemisphere to begin with, and the dorsal pathway just isn't as capable of doing that? So yeah, it's a really interesting question. And yeah, we generally are finding that the dorsal pathway is disrupted here. But I do want to know one thing is that we did do behavioral testing with these patients, and they actually have really good object recognition abilities. I can show you that briefly. So there's like this second level puzzle. Let me see, I think I have that. Yeah. So we have face and word recognition and then object recognition. And you can see that the patients are, lower than the controls here, but they're not failing. Right, like this, they would not qualify for having, prosopagnosia or object agnosia or any of these things. So it's still a question, like given that, particularly dorsal pathway, all these, and they can read and write, just to be clear, they don't have, crazy impaired motor movements or anything like that. So given that neurally it hasn't reorganized, they still have really good ability. So it might be more multivariate, it might be supported by more distributed representation. It's unclear. Yeah.
Can we look at the infant data?
Yeah. Of course. Well, the looking
The sort of
or model.
map.
Oh, yeah. These data.
Neonates. So there are lots of ways you could describe the similarities and differences. And
Yeah.
to me, it doesn't sort of jump out the dorsal's more similar than ventral is.
Mhm.
So I just love to think about what other ways you could look at these connectivity, or like how, like from this, how sure are you that dorsal's earlier?
Yeah. So let me tell you about some of the controls we ran. So, and I should have mentioned this as I was speaking.
So wait, first, what is the
Yeah.
similarity measuring?
Yeah, so it is Pearson correlation between regions, right? And then it's projected into MDS space by taking
We're
the.
not accounting.
So yeah, let me get to that. Yeah, okay. So as Allison's alluding to, there's also these regions are closer to each other, and in fMRI there's might be spatial, like a spatial autocorrelation, such that adjacent cortex might be more similar no matter what. So the very first control that one could do is you analyze the data going across hemisphere. Basically take, one region in the left hemisphere and then correlate it to every other region in the right hemisphere. And that's how you build your similarity matrix. So when you do that, we actually find the same organization. It doesn't make much difference. Correlations are weaker, but it's still there. The second thing you can do is you can just regress out distance. And again, that also holds. And then for these data, you might ask, well, maybe the signal is better in the dorsal pathway than the ventral pathway. The ventral pathway is more medial, it's further from the coil, so maybe it's just noisier. So what we do is we compute a noise ceiling for each of these networks at each individual. Basically ask how reliable are these networks for every individual, and then we normalize these correlations by that noise ceiling. And we find the same exact pattern. You can, I've crossed it 10 other ways. So it doesn't seem to be explained by the specific, and it is, so I will note, it is true that the signal is better in the dorsal pathway, ventral pathway, but even when you account for that, you still find this pattern. Yeah. But it's a really important consideration with neonatal data. Like,
So I'm interested in this contrast of local and global structure for objects, and given your the indications here of a earlier maturing dorsal pathway, as well as the EEG temporal asynchrony, I'm wondering how much of this is mapping onto a broader developmental story of magno versus parvo
I
maturation.
think 100% is. Yeah, I absolutely think that the precursor to this is that the magnocellular pathway is also more mature, and there's evidence of that. That even infants, they're more sensitive to magnocellular information than parvocellular information. I absolutely think that's the precursor to this. Yeah, and I don't find it inconsistent with this, because ultimately the magnocellular pathway projects preferentially to the dorsal pathway, parvocellular projects preferentially to the ventral pathway. If that's driving that development, that's absolutely fine. Yeah.
That's another question.
Sure.
What's the skeleton look like for a face?
Yeah.
And maybe to embed in that,
Yeah.
are all categories treated equally, or are some more diagnostically special? I'm thinking viewpoint with a skeletal shape, global shape base versus local features. And you push it a little further, is that why we have face patches in the ventral stream?
Yeah. And this is this is related to a couple points. So depends on how you compute it, right? So if you just compute it from the head, the outline of the head, it's like it's literally almost like the T shape to some degree, right? That we talk about being very important. But if you calculate it with all the internal features, you also get the T shape, but with circles around each feature, right? So I don't know anyone that's specifically tested whether that's a good diagnostic of a face space, or at least for configural face perception. I think it might actually work, given that it computes it still maintains where those features are relative to one another. So it might still work for configural face perception, but obviously not featural face perception. And what was the second part of your question?
Well then, given like thinking tools versus faces, tools have a lot more of these like joints and angles to them that seem like there's more diagnostic information in a skeletal characterization versus local feature. So to what extent is this sort of maybe mapping onto a broader categorical distinction?
Yeah, I mean, I absolutely think it does map onto category distinctions, but I think the simplicity or complexity of the skeleton is describing the simplicity or complexity of the object, right? So, a pencil and a stick and all of these other things can have the same skeletons, and we're going to say the shapes are all the same, right? But I think, this isn't to say that there's no role for visual features. In fact, I would argue that often local visual features are plenty sufficient, right? We can still recognize there was a face in this image, even though we scrambled it. And there's this funny car, and like even when you do really low resolution, like that this is part of a glass, part of glasses, this is part of the eagle, right? Like take a really small snapshot, we can actually do recognition pretty well. And this is partially why deep neural nets have been so successful, right? You can't argue that they haven't worked, that there's still gaps between humans and neural nets, sure, but they obviously get really far just using these kinds of local features. So, and of course, for subordinate categorization, right, saying stick versus pencil or different kinds of chair, whatever, those local features become much more important because now the structure is not diagnostic.